All right, it's 11 o'clock. We'll go ahead and get started. So, hi everyone. This is Jason Key from SB Grid at Harvard Medical School. Thanks for joining our webinar today. I just want to point out upcoming webinars. We've got CAVR on June 13th, a tool for analyzing cavities and pathways in protein structure. Then in July, we've got JAWView, a sort of sequence analysis and all-around general tool for sequence analysis and alignments and that sort of thing. We're taking August off, and then ShellX in September. So we've got some cool webinars queued up. Today, I'd like to introduce Stephen Lewis and Jane Richardson from Duke University. So we're very lucky to have them join us for our webinar. Today, they're going to talk to us about correcting structure errors in RNA with Erraser. So Stephen, are you there?
I'm there. We're ready to go. Are you ready?
Yep. Take it away.
All right. You said systems checked. Everything good?
Everything looks great.
All right. So Erraser is an algorithm that was assembled by Residue at Stanford. Here's the publication over here on the right. I've worked a lot with Fang and Perrin on picking apart how Erraser works. And the acronym stands for Enumerative Real Space Refinement Assisted by Electron Density under Rosetta. And what Erraser is a tool for fixing RNA errors. And it works by combining two major modeling packages. Erraser is a part of Rosetta. Rosetta is one of the major macromolecular modeling packages. Until relatively recently, Rosetta has been a modeling-only package, but in the last couple of years, we've added bits and pieces to Rosetta to let it handle some level of crystallographic data to make it useful for refinement. And in Erraser, Rosetta is used for its sampling prowess. Rosetta is very good at sampling lots of different structures, and so Rosetta is used to sample different structures and find different conformations that correct errors. Phoenix is a major crystallography package, and Phoenix is used to detect where the errors are and validate that the errors have been corrected. And another way to look at it is Erraser itself runs as part of Rosetta, but it fits into the Phoenix pipeline of taking your data from your crystal data all the way to your finished model.
One other point, it actually involves three packages, and the third one is Molprobity, because neither Rosetta nor Phoenix would know how to detect errors without Molprobity.
Yes. So what sorts of errors is Erraser useful for fixing? I'm going to hand off to Jane.
So ever since the ribosome structures came out in 2000, our lab has been trying to help understand them better, help model them better, deal with RNA structure, which is absolutely fascinating and has even illuminated our study of proteins. And I imagine that anyone who has chosen to join this webinar may not need this introduction, but I'm going to give it anyway. So this is trying to explain why this is a hard problem, and it hasn't really been solved before, but we think Erraser now really handles this whole issue quite well. So like any other structure, if you can get very high resolution, you can really see everything and you don't need much help. So the example on the left here, at 1 angstrom resolution, you can see where every atom is, and so you know what the pucker of the ribose is, you know which way the atoms zigzag along the backbone. But for biologically interesting RNAs, you essentially never get that level. You're very lucky to get 2.5 angstrom. Usually it's 3.5 angstrom structures of really big, interesting RNAs and ribonucleoproteins. And so on the right is exactly the same piece of the sarsen-ricin loop from the 50S ribosome. And you can see the phosphates still marvelously, since they're essentially spherically symmetric. They show up at all resolutions pretty well. And the bases are big and flat and pretty recognizable anywhere. But the rest of it is really very undistinguished at this sort of resolution. you just see a tube and you don't know, you can't see the ribose pucker and you can't see which way the atoms zigzag. And so the result of this is that at usual resolutions, the bases look gorgeous. They have wonderful stacking shown by these blue and green van der Waals dots, and excellent base paired hydrogen bonds shown by the little pale green pillows of H-bond dots. But if you look at the backbone, it has a lot of problems. Now, this is an unusually bad area, but even at the original Maris mortuae ribosome structure at 2.4 angstroms, about 1 residue in 10 has something impossible on the backbone. And you don't see these until you add all the hydrogens explicitly, which is what Malprobity is particularly good at, and that's one reason that we got into this in the first place. So the other reason for the problem is that there are too many variables per observable, basically. One nucleotide has somewhere between 6 and 9 parameters, depending on what you count. The way we analyze it, we parse the backbone into what we call suites because they go from sugar to sugar or base to base, and they have more internal correlation, and if you're going to get clashes, they'll usually be inside the suite rather than inside the chemical nucleotide. And this has 7 parameters because we use delta as the indicator of base pucker on both ends. And the RNA Ontology Consortium, which was basically everybody who'd been looking at detailed backbone structure, came up with a nomenclature where we have 2-character identifiers, a number for the first half of the suite and a letter for the second half. And we've identified so far 56 valid backbone conformations that we think are real. An RNA backbone, for the most part, is pretty rotameric or conformeric. It has barriers between most of these conformations. And so we end up being able to name a structure by putting this 2-character identifier for the backbone conformation in between the base identities. And with this plus base pairing, you have a very complete description of the structure. So one of the most influential and awkward things about RNA backbone is getting the ribose pucker right. And in RNA, this is not true in DNA. It really is two-state to a very, very good approximation. If you're going to do catalysis, you end up with other puckers, but in an equilibrium structure that you would do crystallography on, even at small molecule level, essentially the pucker is either C2' endo or C3' endo. And this matters because changing the pucker slings around all the things attached to it, so that the 2 directions of the backbone and the direction of the base, and as you can see here, with the base going this way, the directions change enormously. In the 3', they're more or less along the same line here, and in the 2', they're almost opposite each other. So this is really critical to get the pucker right. And one of the key discoveries that we made along the way of looking at this stuff is that there is a way to use what you can see, which is the phosphate and the base, and very robustly figure out what the pucker should be. And that's by dropping a perpendicular from the 3' phosphate to the line of the glycosidic bond from the base, the bond between the base and the sugar. And if it's long, it's more than 3 angstroms, then the pucker is 3', and if it's short, less than 2.9 or so, then it really should be 2' pucker, which is more unusual, but still there are quite a lot of them in any large RNA. So we get a robust pucker diagnosis, and in Phoenix Refine, we use that to set pucker-specific targets, both for bond lengths and angles, which change a little bit, and for dihedrals, which lets Phoenix Refine sometimes fix a wrong pucker, but it usually doesn't work because there's a big barrier between the 2 pucker states. So that analysis is also part of what Erraser uses It runs Molprobity on the structure you give it in order to decide which things to work on. And so it will always work on something that's diagnosed as having the wrong pucker. So, for instance, here's an example from one of the ribosomes where this suite is not one of the recognized ones. So bang bang means that it isn't in our list of 56. It doesn't mean it's necessarily wrong, but most of the time it does, of course. And it has a really terrible clash here. These two hydrogens are overlapping by about an angstrom, which is just not physically possible. And we have done fix-ups of this particular residue lots of ways, manually, which is really, really hard, and with some earlier automated fix-ups like RNA-BC and Arcrane, which is partially automated in COOT. And this is just a really easy one for Erraser. So we were involved very heavily in the initial testing of Erraser, and at this point we're eagerly using it on all sorts of things. And it finally does what we were after, and you can actually fix most of the diagnosed problems in an RNA structure, and it definitely works to probably 3.5 angstroms. We haven't actually tested it beyond that, but there aren't too many structures beyond that. And what it does is you first should do refinement, definitely, and probably Phoenix refinement. That's what it's been tested on, and the Phoenix output is what it's expecting to get, so it's probably worth running a short refinement in Phoenix, even if that isn't what you've been doing. Then it runs Molprobity, and it does a Rosetta Relax refinement, and then it does this new stepwise assembly, which is an exhaustive local conformational search. It's a pruned one, but it actually looks at all the possibilities, which is sort of heretical for Rosetta, but anyway, that's the way it works.
It's heretical, but Reju keeps finding things that work, and we keep listening to him.
Yeah. Then it does the Relax again in Rosetta, and then you want to put it back in refinement, and again, what we've tested is putting it back into Phoenix, but at any rate, you definitely put it back in refinement. And you get some spectacular results. This is a Molprobity summary from actually a quite nice structure, a small 1.9 angstrom riboswitch from Rob Beatty's lab. And this is an old one. His newer ones are better than this, but still have problems. And the yellow and red, does that mean? No, that's us.
It's not
Sorry.
interrupting us.
Sorry, it's our own phone, not yours. The yellow and red mean areas where we think there are more problems than there should be. And you note that there's really terrible geometry here. And this has been one of our frustrations with RNA, that it's very difficult to make the geometry really behave as opposed to a protein. Afterward, in this case, it's almost perfect. And it gets near perfect in most cases, so everything is green now. And note also the R-free goes down some. So to look at individual examples, here's a GNRA tetraloop in a different riboswitch, actually, which is going to be our main example today. It has these red spikes for clashes, all atom clashes, bad geometry, particularly here on this ribose. This one doesn't have any actual pucker outliers, but it really looks a lot better afterward. And then my favorite place in this structure is a non-contiguous contact between different pieces of the structure right next to a magnesium binding site. And here we have two pucker outliers that are shown by these magenta crosses and lines, clash outliers, and a lot of bang-bangs for backbone conformations. And although Erraser does not see the metal ions at this point, and it has individual torsion angle targets, but it doesn't explicitly use our legal list of 56 conformers, but it almost always comes up with them. which I find extremely impressive and kind of surprising. Anyway, this is a really lovely fix-up, and it has better ligand geometry to the magnesium even. So I'll let Stephen tell you how to actually get all this wonderful stuff.
So now that we know something about why you'd want to use Erraser, as I said, it's part of Rosetta. Rosetta is primarily a command line interface program, and so Erraser is primarily a command line interface program. Fortunately, it's pretty simple to use. In broad strokes, you need two inputs. You need a PDB formatted model containing your RNA structure, and you need a CCP4 formatted electron density map to go with it. The electron density is just so that Rosetta knows not to drift too far from your observed density when proposing geometrically better solutions. The output that you get from Erraser is a corrected model in the PDB format, which is ready to be re-input back into your refinement pipeline. The PDB itself, for kind of using this to make your new structures, you'll be doing this in the middle of refinement, so you'll have a model that's being output from your crystallographic refinement program. We suggest Phoenix because it's what Erraser is expecting. I suspect other programs will work, although there may be some corner cases that need a little help. I will note that Rosetta is going to use a full atom score function. It absolutely requires the hydrogens, and it's going to add the hydrogens if you don't. It also, Rosetta removes atoms that it sees that have 0 occupancies. At this time, Erraser can't treat alternates, and it can't treat non-canonical modifications to the RNA bases. So just beware that those are limitations at the time. We're definitely going to be working on non-canonical bases, although having a treatment of alternates is a bit further in the future. Your electron density map, it needs to be CCP4 formatted density map. And for best results in refinement, we suggest removing the R-free reflections to reduce overfitting and fill in the missing data with calculated data to avoid truncation artifacts. This kind of the step before you hit Erraser is going to be refinement. I'll just briefly take a look at what that would be in Phoenix if you were using Phoenix. So in Phoenix, you'd be using phoenix.refine. In phoenix.refine, under the Output tab, there are options for, after refinement, writing out electron density maps in CCP4 format. So you'd click this box here, and then you'd click this box here to bring up this Phoenix dialog. These are the options that we suggest you change away from their defaults for best results with Erraser, highlighted here in red. And these are basically the things that I just talked about. So now that you have your inputs put together, you've got a PDB and you've got a map, how do you use Erraser? The easiest way to get a quick taste of Erraser is Rosie. Rosie is the Rosetta online server that includes everyone, which is put together by my good friend Sergey and Jeff Gray's lab at Johns Hopkins. And it's great for doing a test run and for running smaller structures, just to get a feel for the program. And so let me break out a presentation and show you what it looks like on the web. Let's see, we're still sharing. Yes, good. So this is the Rosie website. It does a lot of different Rosetta. It accepts runs of lots of different Rosetta protocols you might want to run. We're talking about, of course, about Erraser. So when you click on Erraser, you have the options to submit Erraser tasks. There's a server queue and there's documentation. The documentation I strongly suggest taking a look at if you want to use it. But since we're talking about how to use it right now, we don't need to. This is what we're talking about, is documentation basically. And then actually submitting a job is pretty straightforward. You've got a couple of boxes to describe what the job is going to be for your own use. You say that you want the standard Erraser protocol, you give it your PDB, you give it your map, you tell it the resolution of your map, and you tell it what, if any, residues you would like fixed during the refinement. I'll circle back around to fixing the residues later in the talk, but there's some important ideas for why you would want residues fixed. And then the other thing, once it's run, this is what the output looks like on the server. It tells you there's some like server stuff of how long it took, blah, blah, blah. You can download your final PDB model here, and then it also offers some statistics on what Eraser did in this particular run. Okay. I had slides in case the internet wasn't working, but we can skip through these since we just looked at it. So if you run a test of Eraser on Rosie and you know you want to run it in a more powerful mode, then you can also run it locally or using SBGrid. If you're going to run it locally, it's similarly pretty simple to get started. It's a command line interface. Eraser.py is the script that runs Eraser. It takes an argument, what PDB am I working with, what map am I working with, and what's my map resolution. And so you run that command line, and you get a little bit of text output, and of course, and then at the end you get your completed model. I will warn you that when you run this, you should expect about this much to come out very quickly because it's just basically the setup steps. And then it'll get to starting whole structure minimization 1, and it may look like it's hanging. It's not hanging. It's just that it's capturing all the Rosetta output and not outputting it to the screen. But don't worry about it if it looks like it's sitting here for quite a while. This particular run, which is a complete refinement of 2GIS, took several hours. It takes several hours on a single processor. This is a pretty slow processor for today's computers. So you can expect it to run faster on a newer machine. Unfortunately, at this time, Eraser does not run, and there's no multi-threading capability. There's no parallel capability. So you do have to run relatively long single runs rather than parallelizing. That's something that we're going to be working on. So this is what 2GIS looked like before we did refinement. You can see lots of clashes. In this red color, you can see lots of geometric outliers in a variety of places in the structure. And this is what Eraser does with it. There are just a handful of clashes left, and all the geometric outliers have been eliminated.
So again, one of the neat things about this is that we've discovered, we think the reason the geometry is so hard to get ideal for RNA is that there are things fit in the wrong local minimum all over the place, like incorrect puckers and backbone twisted the wrong way. And that once you fix these, as Eraser can do, then you can make the geometry ideal, and it will stay that way in refinement. And so we don't worry about that part of it anymore. And it also means that if you have intractably bad geometry, you should worry about more major fix-ups first.
So that gives you an outline of doing a global run with Eraser. The Eraser.py Python program, it takes several other options for things you might want to do. There's a couple of options for file organization. There's -out PDB, useful for putting your output in a different place or with a different file name. This is particularly useful if you're doing groups of residues refining separately and you want the outputs to go to different file names. Kept temp folder keeps all the files that Rosetta generates during the run and keeps them. Under the hood, Eraser is running lots of short Rosetta runs. This flag keeps all of those if you wanted to look at exactly what Rosetta was doing. Use existing temp folder. This is an automatic behavior, but there is a flag for it. This is useful if the run gets interrupted. It can automatically pick up from whatever Rosetta step it did last. So if it does 30 Rosetta steps in the middle, it can pick up at the 15th where it got interrupted. Iteration is a very important option that you have when running from command line. You do not have this option if you're going to run it on a server. So this is the big argument for using it locally or on SBGrid resources. The inIterate flag runs multiple iterations of Eraser. Eraser Eraser's corrections will suggest or allow further corrections, and so running it in an iterated fashion will get you a better model than running it only once. We suggest trying 3 iterations. That seems to be about where the time versus improvement trade-off is, but it's of course 3 times more expensive. I will say that it's pretty much equivalent to just resubmitting Eraser output as Eraser input. So if you choose to run 3 runs sequentially rather than using the iterate flag and check for yourself in between runs if you think it's good enough yet, that will give you the same data. Some other options, there are a series of options for residue selection. Residue selection is an important idea in Eraser for a couple of reasons. First, Eraser uses Phoenix and Molprobity to automatically detect where it thinks the problems are. And you may wish to override Eraser and tell it where you think the problems are by specifying certain residues. And you can do that with this rebuild extra res flag to force refinement of certain residues. The flip side is the fixed res flag to say, do not refine these residues. The primary reason you'd want to avoid refining residues is that Eraser doesn't see anything but RNA inside the Rosetta runs. It keeps things other than RNA in your structure, but inside the actual Rosetta refinement, the only thing that's present is the RNA. And so if you have RNA very close to an ion or a ligand or protein, they're not actually present during Eraser refinement, and sometimes the protein density will suck the RNA into the protein density, and you'll get impossible answers. Right now, the best way around that is by using the fixed res flag to specify don't change those residues. Actually, the topic of my postdoc is going to be in getting protein and RNA working together in the longer run. Here's an example of what the argument looks like for fixed res and rebuild extra res. It's a chain ID followed by a digit, and also you can run regions of digits when they're all contiguous to make that simpler. Now, the other thing that you can do, since you have control over what residues to change and not change, is you can do just local refinement. Local refinement is much, much faster than global refinement because you're changing fewer residues. There's much less computation to do. Here's an example for a particular set of 3 residues near the active site in this 2GIS structure that we've been discussing. This is the command line that you'd use here along the top. All we've done is added here I added fixed res, and then I fixed all the residues other than the 3 that we're interested in. And this is what it looks like going in, and then coming out, Eraser has been able to fix these pretty significant clashes that we see here in the middle.
And also get legal backbone confirmation.
Oh yes, yes.
Because it did change the pucker.
It's fixed the suite too. All right. There's a couple of other advanced options that are available on command line. There's a rebuild all flag, which just skips error detection and just assumes every residue needs to be rebuilt. Obviously, it's going to be more expensive in terms of computer time. Cut point open is a flag that you should never have to use, but it's there in case Eraser incorrectly detects where you have cut points, where you have residues that ought to be chemically disconnected. But if Eraser tries to chemically connect them because it maybe misunderstood your PDB input, the cut point open flag lets you enforce where you think the cut points are. Then native screen RMSD. Eraser normally caps its models at 2 angstroms RMSD from the input structure so that it assumes that your input is mostly correct and it doesn't need to sample too far from that. Native screen RMSD lets you raise the cap on how far it can sample up as high as 10 angstroms RMSD, and any higher than 10 angstroms and it just doesn't even check RMSD at all. It samples freely. Of course, if you have pretty good electron density, it's just going to stay in the density anyway. It can't go but so far because of the density. After you've run Eraser locally, let me re-emphasize that you head back into refinement with your crystallographic refinement package. And just as a particular example to prove why you really need to do that, if you run Eraser on a structure that starts with no hydrogens, For example, Eraser is going to add the hydrogens. This is really Rosetta adding the hydrogens under the hood. And Rosetta doesn't have any idea what B factors are, so it just puts 0 in for all your B factors for all the hydrogens that it added. This is obviously not something you want in your final structure, so you do need to use crystallographic refinement tools after Eraser to produce your real final model.
So a summary of what Eraser did for 2GIS that shows some interesting things. It also shows comparison with 2 other PDB structures. So there are 3 lines, 1 for GIS and 2 for the other 2 structures, and then the lower ones are the Rosetta output for those 3 structures. They're all very closely related. And here in the input, the pink things are problems in the original structure of various sorts, and they go blue and get corrected very thoroughly. Here's, for instance, well, I'm not going to show you that until later. Anyway, there are a number of nice places that it did corrections and a summary of how it did it. And so out of about 25 or 30 problems, it corrects almost all of them. So they go down to blue in all of the outputs. Then one other amusing thing is that there's one place where it insists on keeping what looks like a bang-bang, but I've colored it green because Eraser has convinced us that this is a legal structure. It's a 1 residue junction between 2 helices, and it fits the density beautifully. It has no outliers, and we think it's real, but it's only occurred 1 time so far in the PDB. So one of the places in here that I thought was amusing is a kink turn, where the original structure had a lot of conformer outliers, a pucker outlier, and some clashes. And essentially everything gets corrected. There's 1 remaining outlier, and if you run this on one of the other structures, it takes this one also into the canonical conformation for a kink turn.
So we're getting, that should give you a nice overview of what you can do with Eraser. Just to remind one more time, currently Eraser only works on RNA, and it only works on the part of your structure that is RNA. The fixed res flag is useful to lock down things that shouldn't be moving. We're currently working on extending Eraser to work in the context to work on protein as well as RNA, which will allow us to treat ribonuclear protein so it can really attack the ribosome. And we're also working on improving it to be able to treat errors that are distant from each other and independent from each other, treat them in parallel, because today's scientific computing resources are, of course, highly parallelized. If you try to run Eraser and you need some help, there's some resources available. On the Rosie web server, there's a documentation page that we looked at briefly. Just for all broadly help with Eraser and with Rosetta in general, we run a forum at rosettacommons.org/forum. I'm more or less the guy in charge of the forums, so if you post a question there, I should be able to help you. And then Phoenix also hosts some useful documentation on running things with Phoenix. That brings us to the end.
Great. Thank you very much. I had a couple of questions, and maybe you can touch on these in relation to RNA structures that also have protein, but it would seem at some point you could re-refine all known RNA structures, because it seems like this is universally applicable to those. Is it something you've done or something you have plans to do?
It's something we have plans to do.
Sort of.
Yeah, well,
I don't know about everyone.
Maybe not all of them. Part of what I intend to be working on in the near future is it's already pretty automated at the running Eraser on 1 structure level. But what I'm hoping to do is, particularly by parallelizing it, is making it easy to run whole structures through in a way that works with existing scientific computing resources, so that it would be easy to re-refine structures of really any size, including the very large ribosomes.
But it's still not perfect. I think we could run everything and post it separately, but I would want to look very carefully at the output by hand before putting them back in the PDB. There were over 20 structures run with the Erraser paper, and I did look at most of them by hand, and in general I think they're wonderful, but as you get to lower resolution there's some problems still. It doesn't necessarily do the right thing at the ends of loops where the structure disappears, and it has problems with, as Stephen said, with modified bases, and we probably will find some other issues. What we want to do as a first step is come up with a really accurate reference structure for each of the major ribosomes, and we think maybe now that's possible. It's something we've been wanting to do for over 10 years, and I think maybe we'll be able to in the next year or two.
Sounds great. So if anyone else has questions, feel free to chat them to me. If I don't see any in the next few, I will assume we'll wrap things up. Looks like everybody's, so far that's about it. Looks really exciting.
I could make one more comment that we have earlier and are now working with other RNA crystallographers to do things with their particular structures. So at the moment we're working with Jennifer Doudna on her HTV structures, and some of the riboswitch work was done along with Rob Beatty. And again, we're collaborating with ribosome people to work on their specific structures. And I think that adds a lot. That's one of the reasons why I'm uncomfortable with just re-refining everything blind, because the people who did the structures know things about them that we don't know.
It's automated, but it's not that automated.
Well,
sure.
Automation is never perfect.
Yeah.
Right. But at some point, this as a tool could allow people to refine at resolutions that presumably they wouldn't have been able to refine at before, or at least in a time frame that was sort of reasonable for a postdoc or a graduate.
Yeah. Well, it certainly lets you get a reasonable structure at a worse resolution than you might have managed before. It really needs an input structure, and so at the moment it doesn't help you with initial fitting. A lot of people are trying to work on that part of the problem, and we are at Phoenix among others. So eventually that'll probably be automated as well, but it isn't yet.
Oh, great. Well, with that, I think we'll wrap up here. We can continue with some details offline by email, and I'd like to thank you very much for presenting today.
Thanks for having us.
Great. Bye.
And let us know what works.
Will do.
I don't know if I just hung up or not.